Kita nak menerima maklumat. Kita orang ada sampai sejum.
Hai, kita dah jumpa viewers species.
We are at the Lake of Flamingo. Yeah, and we have done the task. We got the five here. Oh, we cut it and paste it. And we are done. Yay!
Guys, sekarang ni kita dah sampai ke taman botani. Kita nak buat kuadrat. Okay.
Guys, so we have selected the leaf from the Leptisiospermum tapos, or we can call it as pokok perah.
There are differences in the mean length between the different set of leaves and also mean of the width. However, there is no drastic difference in those sets. The difference is maybe because the age of the individual leaves. The leaves are from the same species based on their similar appearance. The size of the leaf grows with its age. Therefore, the younger the leaf, the smaller its size. The leaves are randomly divided into 3 sets of pens. However, it was arranged into a similar size, causing the mean value was not to differ much from their individual value. Variance is the measure of the spread or dispersion of set values in a data set. It quantifies how much each number in the set differs from the mean, which is the average of the set.
So, the statement meaning is implies that the leaf being referred to exhibit variation in both width and length. In other words, the measurements taken for each leaf differ from one another, suggesting diversity in the physical dimensions of both width and length among the leaf in question.
Okay, and then the standard deviation meanings is measure of amount of variation or dispersion in a set of values, while variance measures the average square deviation of each data point from the mean. Standard deviation is simply the square root of the variance. It is necessary to have different set of measurement for leaf, width and length because to ensure a consistent result. It is hard to determine whether the results are correct if only one set was used. By having different sets, we can compare our result with the other set for consistent result. It is also obeys the law of nature because there is no similar individual in a population.
Quadrat is a defined area of a specific size that is marked out for the purpose of sampling and studying the organism within it. It is a square and rectangular plot demarcated on the ground to standardize the area being observed. So, this is the square that we have plot. Hi guys, so this is our second quadrat, which the area of 7 times 6 meter. And this is the area. We have 4 species in this area. Okay. There are 2 basic requirements for quadrat techniques, which is the area of the quadrat must be known and the pop in the quadrat must be determined exactly. So one of the advantages of using a larger quadrat to survey the sedentary organism is efficiency, in which larger quadrats can cover more area quickly, making them more efficient for sampling larger population of ecosystems. Whereas for disadvantages, data collection complexity, in which analyzing data from larger quadrats may be more complex, especially when dealing with spatial patterns and interactions within the quadrat. One of the advantages is smaller quadrats may provide more precise estimates of population density and distribution within a specific area. For the disadvantages, limited representativeness. The use of smaller quadrants may result in underestimating the overall population or missing key special patterns within the ecosystem. There are various issues concern ecologists in conducting the sampling method. One of the issues is sampling bias, which the logistic of sampling large areas may lead to bias sampling. For example, researchers might be more likely to sample areas that are easily accessible, resulting in an underrepresentation of certain habitats or conditions. In result, this bias can affect the accuracy of density estimates. Other than that, the issues also include detectability and visibility. In large forested areas or aquatic habitats, the organisms of interest may be cryptic, elusive, or have behaviors that make them difficult to detect. This can result in underestimation of true densities, as some individuals may go unnoticed during the sampling process. Last but not least, scale-dependent processes. Ecological processes and interactions can vary at different spatial scales. The scale at which sampling is conducted may influence the observed patterns and relationships, and failing to account for scale-dependent processes can lead to misinterpretation of ecological dynamics. There are various factors that would have caused difference in the number of plant species in the quadrant. One of the factors include habitat heterogeneity, in which the quadrant may cover an ecologically diverse area with varying soil types, moisture levels, and microclimates. Different plant species have specific habitat preferences, and the presence of diverse microhabitats within the quadrant can lead to a variety of plant species. Other than that, succession stages. The area sampled may look at different stages of ecological succession. Early successional stages may be dominated by pioneer species, while later stages might have a more diverse community. The timing and frequency of sampling can influence which stages are captured in the quadrant. Next, spatial variation. Plants may exhibit spatial clustering or dispersion patterns influenced by factors such as competition, fertilization, or dispersal mechanisms. The presence of certain species may positively or negatively influence the establishment of others within a quadrant. If the quadrant captures a specific spatial pattern, it could result in the dominance of certain plant species in that particular area. Environmental variance, such as changes in elevation, temperature, or light availability, can influence the distribution of plant species. The quadrant may span such gradients, leading to different plant species adapted to specific environmental conditions. Last but not least, species rarity and abundance. The quadrant may include areas where certain species are rare but present, while others are more abundant. The combination of rare and common species contributes to the overall diversity observed.
Orderan yang dikaji.